and I look very much forward to giving you some new information on the use of probiotics and diet for some of our really common cat diseases. I was totally amazed by Sandra as well as Galia's lectures today. It's a very hard act to follow. But I'm also very excited to be able to do a few lead-in things for our next speakers as we go through the weekend with Jody today and Dr. Quimby tomorrow. I've gotten to work with all of these people for a long time. You probably don't know, but this is actually gray hair, not blonde. It has been great to be with the people from Purina, especially I'm excited about the Institute because it's really great to be able to do science talks and not talk about products. I have in my center done many industry-sponsored lectures and projects, and I'll be honest, Purina is my favorite overall pet food company for doing research. They are so professional and have such a strong animal care and use committee. They're amazing. But you always want to say nice things about people that bring you to places like Bogotá and buy you nice dinners, and I think we're going to the mountains today. But bottom line, even though they do those great things for us, what we try to do in the lectures is only talk about science. And how I avoid having conflict working with all these different companies, including Purina, is I call it being slide worthy. It won't be on a slide unless they've published something. And that's one of the things that I feel the most strongly that Purina in our country, in the United States, has done many more publications than any of the other pet food companies. And so it's very easy to be able to fill 2 50-minute lectures, mainly with published work. I don't work for Purina directly, and so we will talk about other published studies as well. So this isn't a Purina lecture, but it's great to be at a Purina event. And hello to the 5,000 people I hear that might be watching online today. So don't those cats look familiar? Yeah, no matter where we're living, LATAM or in the United States, we're seeing all of these things. Diarrhea we'll talk about in the second hour, but Giardia, very similar in LATAM and the United States. Cats with herpes, chlamydia, mycoplasma. The uveitis cat in the middle on the bottom. The strains of toxoplasma, you have the strains of feline infectious peritonitis. Basically, we're all the same. And of course, one of the best things that's happened in the last 5 years in feline medicine is actually having drugs to treat the effusive and dry form of feline infectious peritonitis on your lower right. So what I wanted to do today is start with a discussion of these infectious diseases, each of which I believe if I could turn on the cat's immune system, that could actually be a benefit. We're going to have to treat many of these with antiviral or antibiotics for bacteria. But could we make the responses even better by stimulating the immune system? And then if you think about cats and the great lecture that Sandra just gave, stress is a huge part of their life. It's stressful being both a predator, trying to eat and survive, as well as being a prey. Very stressful. So diarrhea, urinary lectures, which we'll have this afternoon with Jody, as well as the respiratory type things that my group has specialized in. I wanted you to meet my family. I know I've met many of you over the years in different lectures in LATAM, but I have some new additions to share. And those of you that I'm just meeting this visit, I do have a wife. I really do. One person likes me, which is good. But I wanted to show you my 5 cats. These are the ones that I left yesterday when I came to Colombia. But guess what happened to their life? It became very stressful 6 months ago because the little Labrador cross named Ladybug Lappin, she arrived with 2 broken legs from the shelter. And so my program, Saving Animals in Shelters by Teaching, that Purina is one of our sponsors, we were able to fix her legs. But no one was adopting her, so she came to our house. The little dog that's a tricolored border collie, that's Jalen Lappin, and she's just arrived from Scotland. And so my wife and I and Sydney, the old Labrador, we're all very happy to have Ladybug and Jalen. What do you think the cats think? Yeah, they're not so happy, especially Jalen, the border collie. She wants to herd those cats. She thinks they're sheep. And so life has been upended a little bit at our house. And so now I'm having to think about how to keep them from being stressed and maybe stimulate their immune system so they don't have a recurrence of things like herpes. So we're all living this at work and in our mixed families with dogs and cats. Do you agree that some cats are hard to give oral drugs to? That's kind of a stupid question, right? They all think that they're a cheetah in the African wilds. And that cat, Kieran Grace, she's now 19 years old. I have never successfully gotten a blood sample from her. 19 years. No way. Without heavy sedation, it's not going to happen. So having to give her an oral treatment that I had to open her mouth and put it in her mouth, I would probably die of cat scratch fever. So what my group, again, I have a large research group at Colorado State. Dr. Quimby was actually part of our team a long time ago. I'm so proud of her. She's so famous now. Bottom line is, if you could feed something that they actually would eat, right, then that would just be fantastic in feline medicine. And so that's why we ended up studying things like diets and probiotics in my research group. So what we'll do in this first 33 minutes, I'll probably go about 31 more minutes before questions, is just a few hints in the stress and the immune stimulation space. Second hour, we'll talk a little bit more about diarrhea specifically. So just to get us on the same page, prebiotic fibers make the good bacteria happy. The probiotic is going to be a specific bacterium that has a benefit to the host that we're going to have to give exogenously. We have then the combination of those two, which would be the symbiotics. And then maybe in 5 years, maybe 10 years, maybe we'll spend more of our time talking about using postbiotics, because wouldn't it be great if we didn't have to keep the organism alive? Yeah, that's one of our problems getting probiotics shipped around the world is they have to live. It has to be a live organism. So as we learn more through these great researchers at the different companies, if we can harness those postbiotics, that would make it a lot easier. So I think you know there are many great pet food companies in LATAM as well as in the United States and around the world. So we're not going to pick any favorites today. But what I do know from working with people like Dr. Gagne that you met this morning, these nutritionists, and there's a review article at the bottom by Ebenezer, these nutritional immunologists, they're always thinking, what can we add to our diets to actually improve things like immune function? And as you all know, having good bacteria happy, those prebiotic fibers by itself, they're actually immune-modulating, usually on the good side. We've now known in dogs and cats that adding bovine colostrum is one of the protein additives to diets, can be an immune stimulant. And in several of the canine diets around the world, we now know that triglycerides also can be beneficial. So yes, this is very clearly defined now in kittens, as well as the inferior species, known as the puppy. So, no, I love dogs and cats, but it is great to know that we can do this during the production of great foods that can be sent around the world. So let's shift to one of the first studies that I believe was published. Purina had already shown some work looking at this in puppies, a probiotic that's called Enterococcus lactis now, strain SF68. That product we'll talk about in both lectures, and so I won't reintroduce it, but that is a proprietary probiotic owned by Nestle Purina. But we first met some of the nutritional immunology people. I met Ebenezer, Jaleel, and Christoph, three of these authors, at the British meeting. I was lecturing about some cat disease, probably respiratory, and these nice guys, very professional in suits, came and asked me, would you be interested if we had a probiotic that would turn on the cat's immune system? Would you be interested in working with us? And I'm like, yeah, that sounds really interesting. And they gave me a paper that was soon to be published before this one about puppies and turning on their production of things like vaccine-associated antibodies. Very convincing research. Now, think back, that's 15 years. I was mainly using lactobacillus for diarrhea cases, and I was getting it at the human pharmacy. We didn't really have any veterinary products at the time. And so they helped us support Dr. Veer's PhD. She did a nice study that showed that this particular strain of Enterococcus lactis is very safe and did have immune-modulating characteristics with the tools that we had available then. As you might know, we've gotten a little bit better on how we can measure the immune responses now. But after we had shown that this probiotic manipulated the T helper cells, did some good things with the B lymphocytes with antibodies as well. Once we proved that, we actually then did a study to see if that probiotic would help us keep cats that were herpes carriers. So they weren't currently sick, these are just herpes carriers. We wanted to see if we could help keep that in remission. And so this paper's been out for a period of time, so I'm not going to take a lot of the lecture. But when we fed the probiotic to one group of herpes cats, and then we gave the placebo to another group, remember we needed to handle both groups because of stress, potentially waking up herpes. And then all you have to do to wake up mild herpes signs is to change them from living in a gang room where everybody's living together and then putting them into a cage in the same room. So it's very mild. We do that every day, right? We go to the vet clinic, changing their housing so these cats were not tortured in any way. But you can see what happened just changing their housing. The cats in the probiotic group convincingly had less herpes-associated illness. And so we've continued working with Enterococcus lactis SF68. Now we can measure all 11,000 genes in a white blood cell. We can measure all 11,000. You probably know it's called bulk transcriptomics. It's really fancy, but now it's cheap enough, inexpensive enough that we can do it in small animal studies. It's been a great part of the omics world that we can now gather more information. And so this is a fancier way to show that lactis, SF68, was able to turn on many important genes and also down-regulated. So we believe very strongly we can say Enterococcus lactis, strain SF68, is an immune-modulating probiotic organism. And in our clinical study, before the fancy work, we were able to show it helped keep herpes in remission. And now why we think that is lactis SF68 turns on B cells, which we've now shown in puppies in 2 studies, as well as in cats. And then I just wanted to show you a little bit of the data today on interleukin 12. If you remember that interleukin, in a research study of perfectly healthy cats that were all adopted, we were able to feed the probiotic to one group. We fed the placebo to the other group, which is actually the commercial product, but no bacteria. So we often do placebo trials. And these cats were turning on antiviral cytokines. And more importantly, guys, before I leave this topic, just more evidence that FS68 is an immune modulator, I'm going to draw your attention to the middle chart. The two charts are basically the same, just two ways to measure. So look at the middle chart, and if you look at the tallest bar, that is 100% survival of feline macrophages. So that's in cell culture. So if you grow it on the plate, 100% of the cells are healthy. If you look at the second bar, the lowest bar, that's when we put feline infectious peritonitis, the virus itself. When we put it on those cells, as we know, that's how FIP gets around the body, is it gets into the macrophages. Well, when we put the macrophages on those healthy virus, the virus on the healthy macrophages, it kills them like crazy. So now look at group A and group B. Group A, we pre-treated the virus with serum from the placebo cats. And so you can see it didn't do much. The virus still killed the cells. If you look at group B, the far right side, yeah, that's when we took the serum from cats fed SS68, mixed it with the virus, put it on the macrophages. And so basically proving that something is being produced by those kittens that are fed the probiotic that can actually inhibit FIP virus. This paper will be submitted to Purina for evaluation because they're part of the funding, but just wanted to show you it's probably these types of cytokines that are helping fight a life-threatening virus. Why I thought that was important for you to hear is now that we know that we could potentially boost the immune system, wouldn't you think about taking that probiotic? That's one of the things that, we all age. I don't have any choice. I haven't been taking probiotics as a prevention for myself, but if I knew they were an immune stimulatory, then that's what changed my mind potentially. Think about COVID. You never know when we're going to get exposed to the next person, right? And so I find this to be really groundbreaking research because we now have evidence that we might be able to help cats before they get sick. And that's one of the things I wanted to share with you today. We're doing the enteric coronavirus study now, and we'll be doing a study where we're looking at treating the FIP cats with GS drugs, the new fancy drugs that work about 80% of the time. But what if we could keep them in remission better? Maybe we could have a better response by giving them an immune-stimulating probiotic at the same time. And so that study will be starting soon. All the cats will get a drug because that's standard of care. Half of them will get the probiotic, half will get the placebo. And if I ever get invited back to LATAM, I'll share the results. Hopefully you'll let me come back. So that was the first one half of the lecture. I think that probiotic, believe it or not, there's almost no immune-stimulating probiotic data for cats from any other company. So that's why we could only talk about one. Now, stress is a little bit different. We do have some other strategies to talk about today. What I'm very interested in is keeping cats out of the shelter. I know different countries in LATAM handle this differently, but we have so many cats in shelters in America. They're often relinquished because a family member has allergies. Sometimes they're eliminating in the house, which we'll learn a lot about from Jody in her 2 lectures today. We also have cats relinquished, the ones that really make a lot of nasal discharge, and they're spraying it around the house when they're sneezing their brains out. Well, you don't want to be a shelter cat. They don't all get adopted. And so our team has been very interested. I've just learned so much from Jody, like you guys will later today, of how can we keep them from being in the house, especially if it's an inflammatory disease like feline interstitial cystitis. And so I couldn't do a stress lecture. I'm sorry I'm giving a lead-in for Jody. But honestly, to do stress starts with manipulating the environment, and Jody will give you a full lecture on some of those factors later. I'm stealing from one of her great review articles. But it can start pretty simple. Going to websites that discuss this, if you can read in English. All these are great to be reviewed a little bit more later this afternoon. But I showed you my cat, Karen Grace. She would use that as part of her defense mechanism. None of my dogs can jump up on top of a door, right? And so it's pretty cool. So I think we all have dealt with this in different ways since we've been learning from Dr. Buffington and Dr. Westrop about how to make our indoor cat environments more friendly to their life. And so I think that that's been very important. But I wanted to share with those of you that hadn't met me before, when Dr. McPhail, the bride, my wife, when we came together, we both had 2 dogs and 2 cats, so 4 each species. So we came together now having 8 animals. And that one little black cat, Luke, he was peeing in the house. And so we first gave them the sun porch. Then I made a hole in my house so they could go into their cat house. That still didn't work, and so then we gave them the outside cat run, where he could go outside and eat grass and be happy. He wanted to be an outside cat, which I wanted him to be an outside cat, but I don't want him being eaten by the American wild dog that we call Coyote, and I didn't want him catching feline leukemia from outdoor cats. But once we gave him the cat run, he quit peeing in the house. And then during COVID, I got a little bit bored because I couldn't come and see people around the country. And so I moved that log pile. See the one underneath my bed window? Yeah, so I moved the log pile and made them a climbing gym. I just wanted you to know that I'm a little bit crazy. No, the cat's, it's a fantastic place to live. And I want to know how many of you in Latam at your own house have a cat wheel? Quimby, did you get one yet? No? Anyway, do your enrichment. Hey, another theory. We believe... Yes, I had a weight control problem with my cat, Scout. I tried. I don't know why they like that toilet the best. That's their favorite watering. I have like 3 water bubblers. I have all these water sources, but they like the toilet. That's a fake toilet, by the way. So I wanted to share with you, there's this theory that cats are chronically dehydrated and that perhaps if we could improve their hydration by doing simple things like drinking water better, canned food, which is 70% water, there's this one theory that perhaps that would actually lead to less stress in the cat life. And I'm sure before the weekend is over, Jody and I can debate some of these things. But it's pretty easy to definitely lessen any type of matrix of mucus and crystals in those blocked toms to try to dilute out their urine. But what I wanted to share with you today is I can't wait to hear Jody's new lecture on this, because some of these new supplements that help cats drink more, it can be very beneficial. This is Toby. He was one of those nasal tumor cats. He's my personal cat. He's now gone to cat heaven. But he got the tumor, and we kind of quit responding finally. And so we wanted him to be comfortable for as long as we could. He lived for 3 extra months after the treatment started failing. He lived an extra 3 months just with a supplement for helping enforce water consumption, which you'll learn more about those things today. So I'm not going to steal any more of Jody's thunder, but I want to show you a study where we looked at a high sodium diet compared to a low sodium diet, because I don't believe that's in Jody's lecture. So as you might know, there are some great urinary stone diets around the world now. We published one showing that those kitties that were little small ones, they'd probably dissolve about 40% of the time if they're struvite. And then if they don't dissolve, they probably have something that didn't dissolve. And that's what we showed. But when we finished the study and showed that that diet was very effective, the people at Purina said, do you have a way that maybe you could assess whether or not feeding a higher sodium food, again, they're very good foods for dissolving stones in the struvite world, keeping struvite and calcium oxalates from returning. We know that. We've got at least 3 in North America. They're all great diets. But if you're thinking now, I'm talking about, would a higher sodium one do better than a lower sodium diet? And so that's what they wanted me to study. So we were doing this all with shelter cats that were being relinquished. And so we're wanting to fix their stones. And if they happen to have feline interstitial cystitis, we wanted to see how these diets might differ, a high sodium and a low sodium. And so we did the complete workup to make sure this group of cats were most likely feline interstitial cystitis, so no stones, no bacteria. So we did the complete workup for free for the shelters. We then randomized, this is a randomized trial, into the high sodium diet and then the more restricted sodium diet. Both of those diets are available in many countries around the world. As you might know, if you do a cysto on these cats, they're going to start bleeding again. And so to follow the hematuria, we wanted to do free catch, and we wanted to induce a little bit of stress. So we would house them overnight so we could get a free catch urine, so we could calculate when the hematuria went away. And so the study was very easy. It went very well. A number of the cats had to be disqualified because their signs went away or they had some other disease. But what I wanted to share with you today is when we then looked at all the results for the first 21 days on diet A versus the first 21 days on diet B, we were able to show a difference between groups. A little bit unexpected to me, these diets are formulated very similarly, except for one has a little bit more sodium. And so our hypothesis was the high sodium diet would do better, which is what turned out to be. Whether or not that was because of drinking more water causing less stress, we can't say for sure. But it does give us room to look at additional studies. But why I wanted to share it with you today is encouraging cats to drink is all in all a good thing. And I'm very excited about this study that now I might lean towards the higher sodium diets. There's 2, and then 1 lower sodium. But I might, as a practitioner, go with the higher sodium because of that work, which is published. All right, last couple of things, and we'll open it up to some questions. It is really great to see worldwide companies thinking about how to lessen stress in cats. It's great. I don't even have to list the companies. You probably know the names of them. So adding natural mother's milk type things like the alzacazepine to the food at a higher concentration. Maybe adding a little bit more tryptophan, which we know can lead to feelings of general good well-being. It's really cool to see those becoming real. And to me, when I say becoming real, that means those companies are funding research and publishing it, because it's not real until it's a publication. So congratulations to each of these studies. So now I'm like, wow, what if a urinary diet wasn't working? And we'll hear what Jody says later. Well, maybe a urinary diet's not working for interstitial cystitis. Maybe I should be using a stress diet. Or some of these diets do both. They do the great urinary things as well as have some of those stress-relieving peptides and amino acids. So I'm very excited about this space. But let me bring you back to lactis, SF68, for a minute. You know, I think we concluded about 20 slides ago that this is an immune-stimulating probiotic, herpes model. Now, if I have a low-grade herpes, I might put them on a month of this probiotic to see if that improves them. But I was thinking it was because of all of our immune stimulation work. But buried in that paper, nice little table that I didn't show you yet, was we showed that SF68 actually stabilized the microbiome as well. And part of that work, the probiotics working with the immune cells, is immune stimulating. We've shown that. But the other thing I didn't realize is that's actually exactly what happens with stress. We're changing our diversity. We're having a less diverse microbiome. And so this is one of your first papers. If you want to read any of these, we can make them available. Is we might have also been lessening stress on the microbiome with a probiotic. And so I predict that you're going to see many, many more talks of this type in the next 5 years because many people are interested. The pet food companies, because if they can eat it, it doesn't cause a lot of stress, right? And that's one of the nice things about the tasty probiotics. There's one that's probably the tastiest around the world because of its palatability enhancer. But we are gathering more and more data. So this is my last 2 or 3 slides I'm going to share, and then we'll open up for questions. I think Galia will do that with me. But I wanted to show you the cut and paste at the bottom from Dr. McGowan. Full disclosure, I'm on the Purina advisory board, and so we get together every year and we give advice as scientists, and we see what's coming because they like their advisors to know what's in the pipeline. So I started watching the work with Bifidobacterium longum, strain 999, about 10 years ago, I think now. It's been a long time since we first started seeing some of the feeding trials, some of this brilliant work that Sandra just showed you with the cats. They've been really good about that type of work in dogs, beagles, and hounds. And I got so excited when I first got to see it, and you can read the proceedings. And I was also told that this will be a paper soon, which is very exciting. But I was like a school child. I'm jumping up and down with excitement. Please let me do the cat study. Please let me do the cat study. I'm like begging them to let me do the cat study. And they let me do it. So I think mainly just to get me to quit begging, they let me do the study. And so we thought about how could we do that, and we stayed with the same herpes model. You know, it's very humane because there's almost no stress, but it's easy to wake up herpes. It's easy to measure sneezing and things like that. We adopt all research animals, just like Purina. We do not kill animals in research in my group. So to me, it was a very feel-good pathway. Purina did not have these cats. They're my cats from vaccine studies. So this was all Colorado State University. Here's what we did. We took those perfectly healthy cats that had been in a vaccine trial. You could not tell that they were herpes carriers, just like 98% of cats in the field. We then fed them the probiotic or the placebo. We then changed their caging, and the trained observers are generally my 4th-year veterinary students that we train them how to score conjunctivitis, how to score the sneezing. So the punchline. This is the same palatability enhancer that's in one of their other products, the SF68 product, and so we know cats like it. We measured exactly to the gram how much they ate. And I can tell you that the cats love the probiotic palatability enhancer. And as you can see, we showed not just improvement in activation of herpes, so less activation of herpes, but we also showed some behavioral things, and even serum cortisols, which are hard to interpret in cats because once you touch them, their cortisols go like that. We basically showed behavioral as well as keeping herpes in remission. Colorado State has gotten clearance now to publish this. It's been published as an abstract, and so you'll see it here in the near future. But great news is, depending on your country, you do have a known stress-relieving probiotic to add to your repertoire when we're trying to keep cats from having diarrhea, activated herpes, worsening stomatitis, peeing in the house. I can just think of many ways that I could use a stress-relieving probiotic. I hope that was worth your time today. I am right on schedule for about 4 or 5 minutes of questions, and I guess I'm the next lecturer as well, so we should be in good shape. But thank you for listening. Appreciate it.